In pharmaceutical laboratories like the one pictured here, scientists run highly regulated quality control tests every day. They rely on spreadsheet software to process raw analytical data, tracking everything from pH logs to final assay calculations. Looking at this classic Microsoft Excel interface, we see a program built for convenience. Without configured controls, a spreadsheet lacks the necessary safeguards to prevent accidental overriding or unauthorized data manipulation. This diagram illustrates the core rule of good manufacturing practices. Raw laboratory data must pass through a strict validation-required logic gate to confirm mathematical accuracy and enforce access controls before the system accepts the input. Locking these variables establishes complete traceability. This creates a secure, permanent electronic record designed to meet stringent GMP standards. Here we have an animated diagram breaking down the 7-step validation pipeline. It starts with Step 1, the user requirement specification. This phase precisely defines the expected data inputs, specific calculation rules, and required outputs for the document. With the rules set, we move to Step 2, design and development. This lays out the actual mathematical architecture, building complex formulas, macros, and conditional logic directly into the spreadsheet's framework. Next is Step 3, risk assessment. The hazard map overlay highlights potential failure points in red. We must anticipate where a user might accidentally overwrite a formula or enter text into a numerical field. Resolving these vulnerabilities prior to deployment ensures the spreadsheet functions as a defensible, GMP-compliant system with validated internal controls. This animated comparison shows Step 4, testing and verification. On the left, we perform the calculations manually. On the right, the Excel formula computes the result. They must match exactly to prove the automation works. Step 5 introduces security and access control. This requires locking the exact cells containing formulas while keeping designated data input cells open for the analysts. The process wraps up with documentation and approval. Every step we've taken is recorded in a validation protocol, culminating in a mandatory final review and sign-off by quality assurance. Let's test this concept. Take a look at this simplified assay calculation interface. If an analyst needs to run a batch of samples, which of these columns must remain editable and which must be locked down? The rule is specific. Raw data cells stay open, while formula cells remain impenetrable. The system rejects any attempt to edit the final assay percentage, but accepts numerical data directly into the sample weight column. Applying security at this granular level ensures that an inadvertent keystroke cannot alter a validated calculation. This diagram illustrates how validation operates in a real-world QC assay calculation. The automated formula pulls raw data from the unlocked sample area and standard concentration cells, then computes the result inside a locked, verified cell. Beyond locking cells, version control is layered over the entire grid. This maintains an unbroken audit trail, recording exactly who accessed the file and any authorized modifications made to the structure. These technical controls satisfy major global regulatory mandates. Implementing them aligns your workflow with 21 CFR Part 11, EU Annex 11, and GAMP 5 requirements for electronic records. Proper validation mathematically proves calculations and secures raw data. These controls provide the documented evidence required to verify data integrity during regulatory inspections. If you found this breakdown helpful, please subscribe to the channel for more pharmaceutical training content.